Hello everyone. Today's topic of our discussion is urea cycle. Ammonia is a relatively strong base, and at physiological pH values, it is mainly present in the form of the ammonium ion. Ammonia and ammonium ion are toxic and, at higher concentrations, cause brain damage. Ammonia, therefore, has to be effectively inactivated and excreted. This can be carried out in various ways, such as aquatic animals can excrete ammonium ion directly. For example, fish excrete ammonium ion through the gills called as Ammoniotelic Animals. Terrestrial vertebrates, including humans, hardly excrete any ammonia, and instead most ammonia is converted into urea before removal called as Ureotelic Animals. Birds and reptiles, by contrast, form uric acid, which is mainly excreted as a solid in order to save water called as Uricotelic Animals. This ammonia is converted to urea. Do you know why? Well, in contrast to ammonia, urea is neutral and therefore relatively non-toxic. The reason for the lack of basicity is the molecule's mesomeric characteristics. Let's understand this mechanism of resonance. This is the structure of urea. You can see free electrons indicated by red. These free electron pairs of the two nitrogen atoms are delocalized over the whole structure. Free electron moves from nitrogen of NH2 group to carbon of carbonyl group, and then from this carbon to oxygen of carbonyl group, as a result of which oxygen gains lone pair and gets negative charge while nitrogen becomes electron deficient, indicated by positive sign. So lone pair from carbon moves to nitrogen, and to fill the valency of carbon, lone pair moves from nitrogen of second NH2 group. Now what happens next? Nitrogen of second NH2 group becomes positively charged and lone pair moves to nitrogen of first NH2 group. So what is the ultimate effect of this delocalization of electrons? Therefore, electrons will no longer able to bind protons. As a small, uncharged molecule, urea is able to cross biological membranes easily. In addition, it is easily transported in the blood and excreted in the urine. Urea is produced only in the liver. Let's understand the steps of urea cycle with structures. In the first step, from hydrogen carbonate and ammonium ion, with ATP molecules being consumed, in the presence of enzyme carbonyl phosphate synthase, carbonyl phosphate is formed in the mitochondria. This phosphate is from ATP, carbonyl group from hydrogen carbonate, and NH2 from ammonium ion. In the next step, phosphate from carbonyl phosphate moves out and citrulline is formed as the carbonyl residue from carbonyl phosphate is transferred to the non-proteinogenic amino acid ornithine. In the presence of enzyme called as ornithine carbonyl transferase, converting it into citrulline, which is also non-proteinogenic. This is passed into the cytoplasm through a transporter. The second NH2 group of the urea molecule is provided by aspartate, which condenses with citrulline into arginosuccinate. As the carbon of citrulline forms a bond with nitrogen of aspartate, ATP is also used. In the presence of an enzyme called arginosuccinate synthase, to shift the equilibrium of the reaction to the side of the product, diphosphate is removed from the equilibrium by hydrolysis. Bond with this carbon breaks. In the presence of enzyme arginosuccinate lyase, and cleavage of fumarate from argininosuccinate takes place. That leads to the production of proteinogenic amino acid, arginine, which is synthesized in this way in animal metabolism. In the final step, this bond breaks, water is added and urea is released by forming a bond of oxygen of water to carbonyl carbon. This reaction takes place in the presence of enzyme called arginase. In addition, ornithine is regenerated and returns through the ornithine transporter into the mitochondria, where it becomes available for the cycle once again. The fumarate produced in step 4 is converted to malate in the presence of fumarate hydratase, and by adding water, this malate converts to oxaloacetate in presence of malate dehydrogenase. from which aspartate is formed again by transamination in the presence of an enzyme called as aspartate transaminase. Do you remember the Urea cycle? These are the reactions that occur in Urea cycle. That was all about today's video. Stay tuned to sqadia.com for more precise and knowledge-stuffed videos.